This lecture is going to be on the simple question of how do fossils form? Well, fossils form under very unique circumstances and are not common. When we look at a geologic history or a context here, in geologic time in fossils, people originally thought that fossils were placed in rocks by supernatural beings. That later, about 450 BC, a Greek historian suggested that fossils were evidence of past life. And soon after, 300 BC, Aristotle abandoned this idea and suggested that the lifelike shapes grew within the rocks but were inorganic. But after that thinking, during the Renaissance, the idea that fossils represent remnants of past life was once again believed, as Nicholas Steno started the modern field of paleontology, by suggesting that fossils containing rocks started as loose sediment with fragments of organisms that later hardened into rock. So how do fossils form? Well, fossils are the preserved remains of trace organisms from the past. Most fossils form when the hard parts of organisms get buried in sediment, where they decompose slowly, and the rock preserves their shape as the rock lithifies or solidifies around them. Paleontologists mostly find hard remains, bones, teeth, shells, over soft body parts. Hard parts are more likely to remain intact long enough to become preserved in sediment, whereas soft tissue decomposes quickly. And the fossils that we do find are actually composed of replacement minerals, typically calcite, dolomite, or silica. So let's look at fossil preservation. That not all organisms that die get preserved in rocks. In fact, only a small percentage do, because it takes special circumstances for a fossil to be preserved. So here are 4 key ingredients. You have to have death in an anoxic, which is oxygen-poor environment. Oxygen reacts with organisms and breaks them down. Scavengers may even eat the carcass. You also have to have rapid burial. If an organism dies in an environment that has a high sedimentation rate, it may get buried before it rots or is scavenged. Next, the presence of hard parts. Organisms with hard parts, bones or shells, take longer to break down. Soft-shelled organisms, such as jellyfish, decay very, very rapidly. And lastly, the lack of diagenesis, or metamorphism. Diagenetic and metamorphic processes, example recrystallization, can destroy existing fossils. So they have to survive once they become fossilized. So here's a picture here. I have nutrient-rich water, flows into a change here in depth. The sediment's going to fall out. Here, notice at the top, you get this algae that grows, feeds, and dies. It lands on the sediment, and it depletes the water itself of oxygen, and it's a perfect mud that kind of slows the process for creating a fossil. So here's a picture forming a fossil. A fish lives and then dies. It gets buried in sediment. Soon the soft tissue decays and the bone remains. Well, layers of sediment then accumulate around the organism as it gets lithified into rock. And then later the land is uplifted, where then we go and seek it out and we find it. And this is kind of what we see here. Here's a little bit of a fossil, a little bit of an impression. So there's different preservation methods. We typically find casts of fossils. So we'll talk about molds and casts. A mold is the original shape of the organism's form, right? It's pictured right here. This would be a mold. But if minerals then fill that in, right? Once the mold is filled in with other materials that were generally dissolved in the water, the casts form from replacement minerals. And that's typically what we find when we find fossils. There's different preservation methods, include tar pits, permafrost, amber, and trace fossils. So tar pits are thick sludgy material that envelops an organism, creating an anoxic environment. You have permafrost. Frozen ground contains an organism intact, for example, like an intact woolly mammoth. The ice itself, let's just say, inhibits oxygen movement. We have amber, hardened tree sap, where insects get trapped like on Jurassic Park, right, in a picture here. And trace fossils, evidence of an organism's presence in the form of tracks, trails, tunnels, burrows, and even dung, what we'll call coprolite. So in this classroom, I've got a lot of impressions and a few different tracks. Molds and casts of 3D impressions in which the surface of an organism is preserved. Organisms buried in sediment die slowly, and they slowly decompose, leaving the cavity that contains an exact imprint of the organism's shape and size. When this hollow space fills the material, this takes on the shape of the mold, forming a cast. We find trace fossils, which are evidence of living organisms, but not the specimen itself. So impressions are anything that takes on the shape of a once living organism, but contains no organic matter. And here's a couple pictures of impressions. Tracks are trace fossils that provide clues of an organism's activity left behind in some sediment now hardened. And here's a dinosaur track. So 2 primary groups or types of fossils. We have body fossils, where whole bodies or pieces of bodies of past organisms, right? Bones, insects preserved in amber, mammoths frozen in ice. And you can have trace fossils. These are what are left behind that are evidence of the past life. Could be footprints, could be burrows, etc. So here are shells, right? This would be a body fossil. Here's a dinosaur footprint, a trace fossil, and here are burrows. These would be trace fossils. The key to much of this is what is called a preservation potential. In other words, what is the likelihood of an organism becoming a fossil is actually based on a statistical means, kind of a probability, called preservation potential. An organism dying on land has a much lower potential to be preserved than an organism dying in a body of water. Well, here's why. Because above sea level has a lower preservation potential. To be immediately buried after death is uncommon, and land is exposed to weathering. So land is generally an erosional environment. However, being below sea level has a much higher preservation potential than being buried on land. There's a continual flow of sediment after death underwater, and that makes this a depositional environment, not an erosional environment. So if I look at this picture of the fish fossils are relatively common, whereas a T-Rex or a dinosaur is going to be relatively uncommon. If it's above land, it's going to be eroded. If it's below sea level, it just has a depositional environment that's going to be able to cover it up immediately. So why water? Well, because all water flows into bodies of water, right? Could be an ocean or a lake, along with the deposition of sediment. So we find anoxic conditions where we find such conditions. We have a greater likelihood of finding fossils where we have water flowing into a region. So if this is a shore, and here's a kind of a cross-section of depth, you're going to find a different set of fossils at this location, then at this location, then at this location, etc. You just have this kind of gradation of different fossils. And we're not going to get into the names of them, but just realize that you have different life that's living in the water that's going to die and just have a depositional environment that's going to cover it up. So I have this picture of different shark's teeth, different prehistoric shark teeth. So why are shark tooth fossils so common? Well, they have the perfect conditions to become fossilized. The sharks live in a marine environment. There's a lot of them, a lot of sharks, and a lot of sharks go through a lot of different teeth in their lifetime. And when they fall into an area of deposition. When they fall out of a shark's mouth, they fall into mud, and they get covered very, very quickly. This all leads to what's called a high preservation potential. In a typical shallow marine environment, right, we're talking preservation potential here, about 30% of organisms have a high preservation rate, right? They have a chance of being fossilized. They have hard parts or shells. 40% have a low preservation potential. They're either fragile shells or soft parts. And 30% have no sturdy or hard parts at all. And they're very, very rarely preserved. So only of the 30% with a high preservation potential, but only a few of them will die in environments that will actually preserve them. Right? Just because this fish dies, right? Water carries small rock particles to lakes and seas. But just because it's died and it's buried in sediment, which forms new rock, doesn't guarantee that this is going to be fossilized. But only a few of them that get preserved can even maybe be discovered later and studied. There are a few extraordinary fossils. Although organisms with soft parts are rarely preserved, there are a number of locations through the world that at one time had unique conditions that were just right for preserving some delicate life forms. We'll look at the La Brea tar pits in LA. The mammals got trapped in tar seeps were preserved. And when we look at La Brea tar pits here, the fast fact here is that there were no dinosaurs at the time of the La Brea tar pits. Dinosaurs had been extinct for 65 million years. But the tar pits are clusters of tar pits around urban LA. And this tar seeps up from the ground for many thousands of years. And the tar is often covered with dust and leaves and water. And over the centuries, animals that were trapped in the tar were preserved as bones. And the animals that died there were preserved almost intact, fully. Here we see a woolly mammoth. Here we have a Pterotornis, flying predator. Here we have a wolf, and here we have these different beetles, all of which were trapped in La Brea tar pits. We have a location in the Solnhofen limestone, Germany. This is 150 million years ago that there are many rare and well-exposed fossils, including some of the oldest bird fossils, called the Archaeopteryx. And this is a famous picture that kind of bridges the gap between dinosaurs and a common ancestor with birds before it branched off. And we can look at Burgess Shale in British Columbia, Canada. Many soft-shelled organisms, such as sponges, were actually preserved there. Researchers have been able to determine even the contents of the stomachs of ancient creatures here. And this here just happens to be a trilobite, a very common organism that was alive during the time of deposition of the Burgess Shale. So the process of permineralization is a process of fossilization that occurs when an organism is buried. The empty spaces within an organism get filled, and they get filled with mineral-rich groundwater. The minerals precipitate from the groundwater when the water leaves, and they occupy the empty spaces. And this occurs in very small spaces, such as the living cell tissue. Small-scale permineralization can actually produce very detailed fossils. So this process involves the replacement mineralization of calcite, dolomite, or silica. Those are the three common ones. That replacement minerals are contained in the groundwater and crystallize as the water is pressed, right? Pressure and heat are compressed out. And these minerals replace the original matter. So here's just some pictures of shells. Here's a picture of what would happen if an organism gets compressed in here. The original material gets transported out and new replacement mineral comes in. Now, I've mentioned in the past that shale is very good for forming fossils. But so is calcium carbonate. So is anything involving this carbonate structure. Mainly because CaCO3, right, that's calcite mineral, they actually protect themselves. They're shells of mollusks. They're hard coral exoskeletons. That when the animals die, the soft body parts decompose. But the calcium carbonate remains to actually form these deposits on the ocean floor. So here's a nice kind of a picture of what a hard-shelled skeleton of calcite built up would be forming limestone. So the deposits are buried and compressed and form limestone. And imprints of the hard body parts remain intact as the rock is the fossil. And limestone is a large calcium carbonate material that just commonly forms fossils as well, or fossils can be found in. So I mentioned La Brea tar pits before. Here are La Brea tar pits. We can also find it in permafrost. Here we have a walrus skull. Here's a baby mammoth. Here's a jawbone of a mammoth. And then here's an adult mammoth being excavated within the ice itself. So very good preservation material. We can also have amber. How are fossils formed in amber? Well, insects, spiders, even lizards have been found in almost nearly perfectly preserved in amber. So if a fly lands on a tree that might be looking for food, it gets stuck in sticky sap. And as the fly struggles, it becomes more and more entombed in the sap until it's completely engulfed and suffocated. The tree eventually dies and falls into the swampy water in which it grew. Over the course of millions of years, the amber within the tree becomes a coal deposit. And the fly in the amber is polymerized and hardened into amber. And we later find the amber pretty much intact with these insects within them. Another form of fossils I mentioned before are trace fossils. These are indirect evidence of life in the rock record. They could be animal tracks, evidence of movement. Here's some burrows. Here's some more. These are worm burrows. These are probably more insect burrows. And you can even show the tracks, the animal tracks themselves of different movements. The worm burrows, which are called scalithos, are evidence of the reworking of sediment. And last but never least, kind of a household favorite, coprolite, animal dung. And it looks just like what it is. Coprolite means dung stone, is a fossilized feces, animal waste. And no, coprolite does not smell bad. It has undergone fossilization process. So what can you learn from animal dung? Well, you can learn about diets. Coprolites record diet, feeding behavior, and habitat of prehistoric animals. Paleontologists can study the contents of a coprolite to see what one of the meals of the animal consisted of. If it's a plant eater, it can be partially digested plant material, leaves, seeds, bark, and roots. If it's a meat eater, it could have animal material, right? Protein mush, can be crushed bones or even claws in there. So just because we find coprolite, it's actually what's in the coprolite can tell a great detail about the organism that created it.